Dental caries is a dynamic process where carbohydrates, microorganisms, acids, and plaque all interact. Let's begin by focusing on the role of carbohydrates in dental caries, understanding the critical link between sugar and tooth decay. Carbohydrates are essential for caries because they serve as a substrate for oral bacteria to produce acid. Without them, bacteria cannot produce significant acid, and demineralization does not occur. This is why caries is rare in populations consuming unrefined, low-carbohydrate diets. Evidence from isolated primitive societies shows a low caries index. After exposure to refined diets, which include refined sugars, cooked starches, and sticky foods, there is a sudden increase in caries. This leads to a loss of caries resistance due to increased availability of fermentable carbohydrates. Miller's classic experiments demonstrated this necessity. Teeth incubated with carbohydrates showed decalcification, while those incubated with meat or fat showed no effect. The conclusion is that carbohydrates are necessary for acid production and tooth demineralization. The difference between cooked starch and raw starch is significant. Cooked starch produces acid because cooking gelatinizes starch, making it easier for bacterial enzymes to break down. Raw starch produces little acid, thus cooked rice, bread, and biscuits are cariogenic, while raw grains are minimally cariogenic. Sucrose and starch, when incubated with saliva, produce similar acid levels. This means karyogenicity depends more on bacterial metabolism and oral retention, not just sugar refinement. Only dietary carbohydrates present in the mouth contribute to caries. Salivary carbohydrates are not fermentable. Several factors affect the karyogenicity of carbohydrates. These include the frequency of intake, where frequent exposure is more harmful than total quantity, physical form, with sticky retentive foods being more cariogenic, and chemical composition, where monosaccharides and disaccharides are more cariogenic than sugar alcohols. Additional factors include clearance time, where slow clearance increases caries risk, and the presence of other food constituents like fat, protein, and salt, which reduce stickiness and oral retention of carbohydrates, thus reducing cariogenic potential. The core concept of caries involves bacteria, sugars, and the tooth surface producing organic acids, leading to decay. This triad explains why sugar alone does not cause caries, why plaque control is critical to remove bacteria, and why diet modification reduces the substrate and acid. The caries process begins with dietary carbohydrates, which are fermented by plaque bacteria to produce organic acids, such as lactic acid. This lowers the plaque pH, causing demineralization and resulting in dental caries. In summary, carbohydrates serve as substrates for plaque, with refined diets increasing caries. Miller's findings highlight that fermentable carbohydrates and bacteria lead to acid production and decalcification. Karyogenicity depends on factors like intake frequency, form, composition, clearance, and other food constituents. The role of microorganisms in dental caries is complex. Acidogens, such as S. mutans, initiate lesions. Acidurics, like lactobacilli, progress lesions. Other organisms, such as actinomyces, play roles in root caries. The fundamental principle is that dental caries cannot occur without microorganisms. Neither sugar alone nor a tooth alone results in caries. The combination of bacteria, sugar, and the tooth surface is necessary for caries. The understanding of the bacterial caries theory evolved over time. Miller in the 1880s proposed that bacteria produce acids to decalcify the tooth. Later, Clark isolated S. mutans, finding a consistent association with early lesions, which was rediscovered in the 1960s. Initially, lactobacilli were thought to indicate caries activity. However, later understanding showed they are rare in early lesions but common in deep dentinal caries. The conclusion is that lactobacilli are secondary invaders, important in progression, not initiation. Modern understanding assigns different roles to different organisms. S. mutans initiates caries by colonizing smooth surfaces, producing acid and sticky polysaccharides. Lactobacilli thrive in low pH, predominate in deep dentinal lesions, and maintain and enlarge cavities during caries progression. Actinomyces, specifically A. viscosus and A. naselundii, are associated with root caries colonizing exposed cementum, especially in elderly patients. Pit and fissure caries are often caused by a mixed flora due to the retentive anatomy. S. mutans has unique properties. It is highly acidogenic and aciduric, produces insoluble glucans from sucrose, strongly adheres to enamel, and stores intracellular polysaccharides, which leads to prolonged acid production. Sucrose feeds S mutans, which produces glucosyltransferase forming sticky glucans. These trap acids on the tooth surface, leading to enamel demineralization. Also, sucrose is most dangerous because bacteria use it to produce acids and sticky glucans, which retain plaque and prolong enamel demineralization. Different sites host different bacteria. S. mutans on smooth surfaces, mixed flora in pits, actinomyces on roots, lactobacilli in deep dentin. So caries is a microbiologically diverse disease. Because different bacteria dominate different tooth surfaces, dental caries is not one disease but many microbial patterns. Summary of role of microbes in dental caries. Smooth surfaces, pits, roots, and dentin each select different bacteria, proving caries is site-specific and microbiologically diverse, involving an initiation and progression of the disease. The role plaque should be correlated with role of microbes as well. Earlier it was considered that all plaque is bad, but current evidence suggests that certain bacteria in plaque causes caries. Now we move on to understanding how the acids produced by microorganisms result in tooth demineralization. Plaque bacteria ferment sugars mainly to lactic acid, a strong, low pKa acid produced in large amounts, making it the most destructive to enamel. Though the exact mechanism is not known, plaque acids are produced by bacterial enzymatic glycolysis. Therefore, inhibiting these enzymes, such as by fluoride, reduces acid formation in caries. Remember, caries is not caused by acid in saliva, but by acid trapped on the tooth surface. Saliva washes and buffers acid, but dental plaque holds it long enough to dissolve enamel. Dental plaque acts like a sponge and diffusion barrier, trapping acids on enamel and blocking saliva, so localized acid causes caries, not generalized acid. Simple sugars ferment fast, enter bacteria easily, and cause sharp pH drop.
Why does
glycolysis dominate
cooked
plaque?
starch breaks
Plaque
slowly.
is dense and
Raw
low in
starch
oxygen,
barely
so
ferments.
bacteria use anaerobic glycolysis, breaking sugars into acids. Homofermentative bacteria like S. mutans and lactobacilli produce mainly lactic acid, causing a sharp pH drop and high karyogenicity. Heterofermentative bacteria produce mixed acids and ethanol, so the pH fall is slower and less severe. S. mutans produces very high acid, making it the most karyogenic. S. sanguis is moderate, S. mitis low, and actinomyces varies. Homolactic anaerobically, heterolactic with carbon dioxide, explaining different caries patterns like root versus enamel lesions. Acids produced by bacterial fermentation of dietary carbohydrates, especially lactic acid, cause localized demineralization of tooth structure when retained against enamel by dental plaque. The amount and type of acid produced depend on the carbohydrate source, bacterial species, and environmental conditions within plaque. Let us now focus on the role of dental plaque in dental caries. Dental plaque is a soft bacterial biofilm that forms on teeth within 24 to 48 hours. It resists saliva and tongue cleaning, removable only by brushing. Plaque is essential for dental caries initiation, though its presence alone doesn't always cause cavities. Plaque is 80% water and 20% solids. Solids include bacteria, salivary proteins, extracellular polysaccharides, and calcium/phosphate. The acquired pellicle forms first, helping bacteria attach and trapping acids and sugars near enamel. Plaque is microbiologically diverse. Major karyogenic bacteria are streptococci like S. mutans, actinomyces, and lactobacilli in deep lesions. S. mutans produces sticky glucans and acid, initiating enamel caries. Quality of plaque determines caries risk. Plaque bacteria ferment sugars into acids, mainly lactic acid. pH drops to 4.5 to 5.0 after sugar within minutes. Critical pH of 5.5 triggers enamel demineralization, and below 5.0 causes subsurface lesions. Plaque retains sugars, produces acids, and blocks salivary buffers, keeping pH low. Insoluble glucans increase adherence and reduce neutralization. Plaque can be protective or karyogenic. Dental plaque is a structured microbial biofilm that localizes acid production at the tooth surface, leading to demineralization and initiation of dental caries.